Hello students, Namaste. I think you are ready for today's class and you are feeling well. Today you should be with your pencil, copy, book, etc. with you. And today we are going to continue living beings and their structure, that is unit 3 of grade 8 of science and technology. And under it we have finished about different components of a cell, but all the components of the cells we have not finished. We have finished about nuclear membrane, and then cell wall, cytoplasm, mitochondria, and then what? Plastids and their types. What are they? What are their function? And you know that all of them are not cell components. Some we are of covering, some the sap inside it, and only mitochondria and plastids. And under plastid, we discussed about three types of plastid: chloroplast, chromoplast, and leucoplast. And today we are going to today we are going to study about some more components of a cell. And today we are not going to think about a particular question, but at first we'll do an activity. Yes, and that activity is related with our prior work. You have seen this diagram once because you have done about animal cell and plant cell. I used generalized cell, you remember, in which I shown both of the uh, components, that means those components which are found in plant cell as well as animal cell both. Then you were suggested to sketch the diagrams of plants and animal cells separately and with their labeling. So here two cells are shown, and you know that it is plant cell and it is animal cell. And here you have to label different parts of the animal and the plant cells given below. Arrows are given. Just you have to identify what is it and you have to write its name. Here for both arrows are given. What is the common? You have to write here. Like that you have to fill it. Okay, do fast. Okay, so if you have finished, then you compare your work with that labeling. I think you have done cell wall, and you are seeing that cell wall is found in only plant cell. So for such labeling, here you are used dotted arrows which are showing you differences also. And keep in mind which lines are dotted, because that will help you to compare animal cell and plant cell. Okay, so cell membrane both side, yes, correct. Cytoplasm, yes, the sap filled inside the cell membrane, and then yes, they are nuclei and singular nucleus. And here these structures, yes, they are endoplasmic reticulum, and in both of them there are some rounded structures around that endoplasmic reticulum. They are what? Ribosomes. So here it is better to label endoplasmic reticulum with ribosomes. And you know that ribosomes are found at other parts also. They are found scattered in other region also, not only on the endoplasmic reticulum. Yes. Then vacuole, yes, both side, here vacuole, both side vacuole. And then Golgi bodies, yes, Golgi bodies. And plastid, yes, plastid is not common in animal cell. In very particular type of animal cells, this plastid is found. You are familiar with that animal? Can you say what is that? Yes, that is euglena. Yes, but in all other animals, plastids are not found. So plastid is a part of component of plant cell only. Now you are seeing here that star-like structure. That structure is what? Centrosome, inside which two dotted structures, not dotted, you can say bundle-like structures, bundle of wood, firewood like structures are seen being perpendicular to each other. So they are centrioles. Yes. So centrosome is shown here. And both side, what is the structures? Yes, they are what? Lysosome. Lysosomes are found in both of the cells. And last one, that is to label, they are what? That is mitochondria. They are mitochondria. So in this way, we labeled animal cell and plant cell both, and we recalled the different components, different parts of the cells, and side by side we found that what are differences. Many things we have learned from this activity. Now we are going to start about the next cell organelle that is also important, very important for the cell, that is Golgi body. So here it is 2D picture of Golgi body, and that one is 3D. So you have to, it is easy to sketch. You can sketch this type of structure in what? Animal cell and plant cell. And this portion is found towards the nucleus. Remember it. This portion is called as cis phase. Cis phase. So cis phase is found towards the endoplasmic reticulum, or you can say towards the nucleus. And these plates like structures, the older plate like structures are found against the nucleus. They are called as transphase. So that these terms may not be in your book, but it is easy, they are easy to understand. And here this spiral structure, or you can say that curved structure, yes, that is called as what? Cisternae. And inside cisternae, that cavity like structure is called as lumen. Yes, so that is the major component of Golgi body. There are secretory vesicles also. These vesicles, these are the opening of the tube which carry the substances from endoplasmic reticulum to the Golgi body. So in its function, we will discuss more about it. At first look in structure. So in its structure, what is this? It was at first it was discovered by whom? Yes, Italian scientist Camillo Golgi. Some pronounce Golgi also. So Golgi is not so common, but Golgi is better. So Camillo Golgi in 1898 AD. Yes, and parts, I told you about these parts. Not necessary to label the different parts for your label. Yes, for your label only diagram is enough. But few parts, if you remember, you will be smarter. That's why you have to know it. Yeah, made of lumen and cisternae. So I told you that lumen is that cavity-like structure, and cisternae is what? That spiral structure, I can say curved structure, which is made up of two layers. That is made up of what? Two layers. Yeah. So two layered cell organelle. It is two layered cell organelle. You know that some organelles are non-layered, some organelles are single layered, and some are two layered. So it is two layered organelle. Yes, and found near the nucleus of the cell. In diagram also you have seen that Golgi bodies are found near the nucleus. Yes, and there this face is found, or this part is found orienting towards the nucleus, that is called as cis phase, and the opposite part is called as trans phase. So that is also given here. So they are cis phase and trans phase. So main thing is different plate like structures are seen in this structure, in which or in this organelle, different plate like structures are seen combinedly in this organelle, and in it the folding are called as cisternae, and the cavity in between the foldings are called as what? They are called lumen. Yes? Okay. So what is its function, or what are the functions of Golgi bodies? So first function is they are concerned with secretion of mucus. You know mucus? Mucus is found at inner lining of our stomach. Mucus is sometimes blows from nose also, the thick fluid mucus. Such structures are found in our alimentary canal also at inner surface, which protects the canal from the effect of digestive juices. Otherwise, our intestine will be digested if there is absence of this mucus. Okay. So mucus, sweat we know, yes, tears, yes, saliva, and other juices. Different types of juices are produced, are required for our body, which are produced by what? Golgi bodies. Yes. They form cell plate. Students, so the term here cell division is very new for you. In fact, during cell division, one cell divides into two or more. Yes. So here consider it is animal cell. Then in animal cell, the cell divides into two parts by forming cleavage, that is constrictions, cleavage. And in this way, if here was nucleus, so the nucleus divides into two parts. So in animal cell, there is not role of this Golgi body. But if it is plant cell, then after the division of nucleus, here is cell membrane. So division of nucleus, a plate-like structure is formed in between these two nuclei. So this plate-like structure later changes the form of cell wall. This plate is called as what? Cell plate. And this cell plate is made of, or it is made by what? Golgi body. Okay. So it is cell plate. Yes, student? Okay. Now, so cell plate you have understood. Cell plate is a lining in between two nuclei during the formation of new cells. That process is called as cell division. So now I think you are familiar the term cell division. Cell division is formation of new cells by which number of cells is increased, by which we grow, by which new cells, that means organs are also formed in babies. Yes, so that is due to what? Cell division. Okay. So storing and transportation, they store some fluids and they transport from one place to another place. They take help of others, but there is also role of Golgi bodies in what? Transportation. And they work as package, that means packaging medium. The endoplasmic reticulum produces some secretion and that is brought here through the tubes and they pack them like packing machines. They pack them for the particular regions, for the supply of these things to particular region. So packaging of protein received from what? Endoplasmic reticulum. Yes. Okay. And it also forms lysosomes, or you can say Golgi bodies form lysosomes. Lysosomes are other organelles. What are they? How do they work? We will discuss later. Before that, I am going to give you one activity in which you have to compare mitochondria, plastid and Golgi body. About these two you have finished in prior classes. So here diagrams are given to recall something about them and something you can compare also by seeing the diagram. They also hint you the structure, function also you can recall. So just try, you have to write differences between mitochondria, plastids and Golgi bodies. Try to do fastly. Full sentence is not required, just term or words are enough which are differentiating them. Not only differentiation, compare means you can write similarity also. If two are similar, another is different, that point also can be written. Yes? Okay. So now you have to compare your work with these points. Have you written that mitochondria is elongated in shape? You can say elongated shape, here elongated shape, but it is layered. Number of plate-like structures are seen joined together. Yes, so that is first point. If it is not written, you have to add now. Found in all the cells. That means in plant cells, animal cells, both, that is found. They are found. They are found only in plant cells. And they are found in plant cells and animal cells both. Yes. So two similarities in them and one dissimilarity. More in number. Mitochondria is found more in number. Plastids are found more in number. And Golgi bodies are found in less number. Not only a single as shown in the diagram. Single is shown. It doesn't mean that only single Golgi body is found in a cell. Its number may be different. Okay, it is not that means only one or two, many, but lesser lesser comparatively to other organelles. Only that thing you have to keep in your mind. Okay, that is energy producer. It produces energy. What does it do? It helps in preparation and storage of food. Only chloroplast is given here, that shows the plastid. So plastids are concerned with what? Preparation and storage of food. And here it is related with what? Excretion, nutrition, secretion, transportation also you can write. Yes, packaging. So all the works you can write here. Okay. Next one. Mitochondria is found anywhere in the cell, scattered in the cytoplasm anywhere. Plastid also anywhere in the cell. But this structure, Golgi body, is found close to the nucleus because it has to receive some things, some secretions of endoplasmic reticulum. That's why it is found closer to the nucleus and endoplasmic reticulums. Okay. And last one, you may write something different, that also may be correct, consult your teacher. So here it has cristae. Cristae means that structure, folding structure is called as cristae. Here it is called as cisternae. Here cristae, here cisternae. And here they have layers of pigments. They are pigments. Only leucoplasts do not have pigment, but chloroplasts and chromoplasts have what? Pigments. Okay. So you are clear about that comparison? Okay. Now see about lysosome. Lysosome is made up of, in fact, here it is seen that there are number of layers, but all together they form a single layer. So that is outer membrane, and here lipid layer is found below it. Okay. And inside it there are enzymes. Here are number of labelings to which you do not have to remember. Yes, only enzymes, digestive enzymes you can say, because they digest the unnecessary substances found inside the cell. They may be foreign bodies. Sometimes some foreign bodies also enter inside the cell. Yes, so they are also digested, and sometimes some components of the cells be useless. They die, they destroy by any region. So they are removed by these enzymes. They are digested and made soluble in the cytoplasm, which are later expelled out through our transportation system and excretory system. Okay. So that is about lysosome, and lysosome is considered as suicidal bag also. Can you say why? It is because sometimes it becomes the cause of the death of the cell. It becomes cause of the death of the cell. When these lysosomes burst inside the cells, the cell also dies. Yes. So they are called as suicidal bag, and sometimes our body has to do that to remove unwanted cells. Okay. Okay. Now see, it has single layer covering. So it is single layered component found in the cells of liver, kidneys, spleen and pancreas mainly. Not in all the cells, it is not common. And you know that lysosomes are formed by what? Golgi bodies. Yes. Okay. And in these cells they are common because their function is in these organs. Okay. And it has digestive enzymes about which I have discussed. Okay. Now what is the function of lysosome? Its functions are digestion of protein, fat, glycogen. Glycogen is a form of carbohydrate. When we use carbohydrate, the excess carbohydrate is stored in our liver and in the liver of other organisms also. That is glycogen. Not only that, in mushrooms also glycogen is stored. That's why when we eat livers, if you are that means non-vegetarian, you use meat, and then you have felt that the liver is sweeter in taste relative to other meat, and mushroom is sweeter in taste. If you are vegetarian, then you have tested that means vegetarian, non-vegetarian both eat, but vegetarian have not tested the liver, but they have tested mushroom, and mushroom is also sweeter because of the presence of what? Glycogen. So these things are digested inside the cell by these enzymes, and you know that maximum part of our body are made of these things. It is not necessary that those protein, fat, carbohydrate which are eaten by us, they are digested by it. No, these components form our whole the body. So inside the body, when any organelle made of these structures has to be destroyed, this work is done by lysosome. Okay. Next, Autodigestion of cell parts and foreign bodies. Already I have discussed. Foreign bodies, foreign substances which enter with some required substances which are supplied to the cell. If they are not required for the cell, they are destroyed by it. And the cell parts which are not required for the cell, for the body, they are digested by it. Yes. It supplies nutrition to seeds during their germination. So during germination of seed, its role is more there. So lysosome is not found only in animal cell, that is found in plant cells also, and it helps to supply nutrition to the baby plant which has to germinate, which is found inside the seed and had to come out in the form of new plant. Yes, okay. Now next one is vacuole. Vacuole is very simple structure. Just it has a membrane which is called a stenoplast, and inside it cell sap is filled. That means some water substances, some water and water soluble substances are found here, but there are gas vacuoles also. In cells, gas vacuoles are also found, and these gas vacuoles are filled with only gas, not cell sap. Yes, vacuoles are of different types. What are their function? We will discuss later. Has a single layered covering called stenoplast. We have discussed about it. Filled with cell sap, which is a mixture of water, salt, mineral, enzyme, etc. But remember, not in gas vacuoles, except in gas vacuoles. Small in animal cells and big in plant cells. Not only small, but many in numbers also, you have to keep in mind. And in plant cells, they are bigger and less in number. Okay. Found in the form of gas vacuole, food vacuole, and contractile vacuole. Gas vacuole, food vacuole, and contractile vacuole, and these different types of vacuoles do different functions. Different functions are done by them. So in functions we will discuss about it. Yes. So now its function, it balances amount of water and salt. How? By collecting the excess amount of water and salt, if there it is more in number, or there is just you can say double transportation, the required substances are given and unnecessary substances are taken inside. Okay. And contractile vacuoles are responsible for excretion. You have seen contractile, if you remember the structure of amoeba, paramecium, euglena, in them contractile vacuoles are found which appear to form from central region, and they grow and move towards the marginal region, and later they burst out. That means while their motion from center to the cell membrane region, they collect the unnecessary materials, become larger, 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 and when they come at surface, they throw out the unnecessary substances. So in this way, they do excretion. So excretion is function of what type of vacuoles? Contractile vacuoles. Okay, what is the next vacuole? Gas vacuoles are responsible for flotation. In different types of aquatic plants, gas vacuoles are found which make them lighter, which make their body spongy. Yes, and in some animals are also found that there may be the number of gas vacuoles more in their body, in their cells, which makes their body lighter. Yeah, and next one is food vacuole, which stores food and supplies to the cells when required. Food vacuoles are found, you have seen food vacuole also in amoeba, paramecium. Yes, when they engulf their food or their prey, they change in the form of, or you can say they are stored in the form of what? Food vacuoles. They are digested there and provide nutrition. So three types of vacuoles do three different functions, about which the term we have introduced, but I told that we will discuss later. Okay, students. So up to vacuoles we have finished. Now one activity we are going to do in which you have to write similarities and dissimilarities in lysosome and vacuole. So at first try to write similarities. How many, what similarities you find? Very few they may be, not necessary to make table, just in sentence you can write. Well, now see what is the first point. Both are about round. I can say both of them are about round. Yes, both have single layered cover. that is also single layered. It is also single layered, thick and thin, that is there. Next, they are found in both types of cells. They both of them are present in animal cell as well as in plant cell. Yes. So now, what may be dissimilarity? Just try. What are dissimilarities? Dissimilarities means differences. How do they differ from one each other? Okay. So you have done. Have you written? Lysosome digests things and vacuole stores or excretes. Yes, function. On the basis of function, you can differentiate them. Lysosome is complex and vacuole is simple in structure. Yes, lysosome is more complex relative to vacuole. Vacuole just have a single layer membrane inside which either air, gas or some sap is filled. But here are different structures, lipid layer, other layer, then enzymes, different type of enzymes. Yes, so its structure is, our lysosome's structure is complex. Now, lysosome is filled with enzyme and others in a vacuole may be filled with liquid or air. So, not only structure, what is it filled with is also given here. Okay. So, we have finished some cell components. Still we have to read about some more components. So, next one is what? Centrosome. Centrosome and its function. Centrosome is that structure, star-like structure. Yes. And I told you it looks like bundle of what? Firewood. Yes. And they are adjusted perpendicular to each other. Perpendicular to each other. They are centrioles. Yes, these centrioles have to separate during cell division, about which we discussed. Cell division means what? Formation of new cells by their division. So, there is important role of these centrioles and it is found only in animal cells, not in plant cells. Yes, okay. So, at first about structure, it is found in animal cells only. Yes, we have finished. Has centrioles in it, two centrioles, we are familiar with it. Consists of microtubules. So, these structures, or you can say centrosome as a whole, is made up of some tube-like structures of proteins which are called as what? Microtubules. In fact, they are protein. I told you that maximum components, about all the components of the cells are made of protein, fat, carbohydrates. So, they are mainly, these centrioles are made of what? Microtubules. They are protein. Okay. Plays important role in cell division. So, you may say, what is that important role? In fact, their role is to pull chromosomes to different poles. What is pole? What are chromosomes? So, about chromosomes you will study in the structure of nucleus, that may be in detail in next class, but today you will be familiar with chromosomes. Okay. So, it is about centrosome. Now, we are going to read about, now we are going to see about the central structure of the cell, not only central, but it is the main master of the cell. Yes, brain of the cell. That is what? Nucleus. Okay. Nucleus is single in a cell. Usually, in some animal cells that is found two also in one cell. So, there are exceptional cases everywhere. So, generally they are found single in each cell and it is made of four major parts. The lining, that is covering, which separates the sap inside the nucleus and outside the nucleus, that is called as nuclear membrane or sometimes nuclear envelope. Okay. So, identify, just today we will identify the different parts. About function we will not discuss. Then next, inside it the sap field is called as nucleoplasm. In fact, a cell has two types of saps, two types of liquids inside it. One is nucleoplasm, which is found inside the nuclear membrane. Another one is cytoplasm, which is out of the nuclear membrane and enclosed by cell membrane. In between cell membrane and nuclear membrane, there is what? Cytoplasm. And both combinedly are called as protoplasm. So protoplasm is basis of life. Protoplasm is basis of life because each and everything of the cell are found inside it, which are required for different life processes. Okay, so here it is nucleoplasm. Inside nucleoplasm, some thread-like structures be visible at particular condition. Yes, otherwise they are disappeared. In earlier stage of the formation of a cell, they are invisible. They are not seen. Later, they change in the form of network of fibers. They are called as chromatin fibers. Then, when the cell becomes ready to divide into new cells, then it becomes more distinct, coiled, and divides into particular number of thread-like structures. That means they divide into number of thread-like structures, and they are called as chromosomes, about which we discussed in centriole. Chromosomes are found particular in number in particular organisms. You know that what is the number of in our each cell? It is 23 pairs. 23 pairs in vegetative cells and 23 in reproductive cells. So what are the vegetative cells and reproductive cells? You will be more clear when you will be taught about reproduction, about which you have to read in coming lessons. Okay. So all four, nucleolus is one rounded structure. So there are four major parts of it. Okay, students, central part of a cell, two layered covering and consists of four organelles. That means four parts. So it is clear. Now I am going to give you one project work related with animal cell and plant cell. So here one sample is given. You have to make better than it. You have to use any other substance which are used here. So they are model of plant cell and animal cell. Yes, so make a model of animal cell and plant cell using local materials. Yes, no expensive materials you have to use here. Only wastage, useless materials, locally available materials you can use and make to attractive cells with labeling. Okay. Now let's see what have we learned. Today we have discussed about some remaining organelles of the cell like lysosome, centrosome, vacuole, yes, and then Golgi body, nucleus. Nucleus is next part, yes. Okay, let's see what have you understand. First question is, which cell organelle is considered as suicidal bag of a cell? So it is very common, I think still that is in your brain. It is what? Lysosome. Yes, you have done. Okay. Next question. Which of following produces important juices for body? So there are four alternatives. Easily you can guess that they are what? Golgi bodies. So Golgi body is its answer. Okay, students. Now next question is, which of following statement is true about cell organelles? Gas vacuoles help in excretion of a cell. Centrosome is found inside nucleus of a cell. Golgi bodies receive protein from endoplasmic reticulum, or lysosome form cell plate during cell division. So I think you have guessed the correct one. That is what? Golgi bodies receive protein from endoplasmic reticulum. Okay. Now next is related with the table. You have to study the table and you have to match. You have to find out the correct matching out of these four. Okay. So I think you have done. That is D number. Let's see. 1B, yes, animal cell have centrosome. 2D, digestive enzymes is found in what? Lysosomes. 3A, storage of food in vacuole. Fourth one is already correct, not necessary to see. Okay. Now write the answer of following questions. There are some questions to which you have to copy and you have to write answer, and after answering them you have to show your teacher. First question is given here. Write two similarities and two differences in between lysosome and Golgi body. I think you will feel easier because in activities also we have done some things. Name any two cell organelles made of single layer. Yes, you can name. Yes. What is the function of lysosome? Single function is, you are asked here is, not what are. So single function is enough. Which organelle is considered as suicidal bag of a cell? Also describe it in short. Now you have got the answer, very close. Yeah, okay, that is it for you. And here are given three diagrams, A, B, C. Answer the questions based on the given diagram. The question number 1 is, which of them is found at the central part of a cell? Easy? Okay, easily you can guess. Which of them contains chromatin fiber? Same. Yes, as you have answered in number 1. Which of them forms cell plate in plant cell? Cell plate formation. Yes? Yes, it is here. And last one. Which of them has a single layered cover? So, only that is remained, and really that is the answer. Yes. Okay, students. So, that's all for today. In next class, we'll be with some new things. Namaste.